This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 95, recorded on August 13, 2010. Hello, everybody. I'm Vincent Racaniello, and this is TWiV, your weekly podcast all about viruses. Joining me today from New York City, it's Dickson Despommier.
Hey, Vince.
How you doing, Dickson?
I'm doing well.
Sorry
I missed the Bozeman meeting. That was one of my big emotional letdowns by not being able to get out there because I enjoy that area of the world, and I understand your podcast out there went really well.
We had a great time, and next time if we're out there again somewhere else for ASV, maybe you can join us.
I'd love to.
Also with us today from Western Massachusetts is Alan Dove.
Pleasure to be here.
Welcome back.
Yeah.
Down in North Central Florida is Rich Condit.
Hi, everybody.
Hey, Rich.
Great to be here.
And we have an in-guest studio. We have an in-studio guest today, Michael Zigman from the New York Academy of Sciences. Welcome.
Good afternoon.
First in-guests in-studio. Why can't I say it properly? The first in-studio guest we've had.
Well, you know, an in-guest studio would give you great sound fidelity.
That's right. Studio in the guest. That's right. Today we are back to talking about different stories in virology. We've had a run of unusual TWiVs lately, and this is the old format. And, of course, we have a bunch of them as usual, and we have to start out with this week's announcement by the World Health Organization that the pandemic is over.
Great.
I think great.
This is a pandemic that Dickson said would never happen.
Right, and you can quote me.
And we have, and you're on record. So the Director General of World Health Organization, Margaret Chen, made an announcement this week. The world is no longer in phase 6 of influenza pandemic alert. We are now moving into the post-pandemic period. Mm-hmm. The new H1N1 virus has largely run its course. And she basically says that the outbreaks are very different now. They're occurring in season rather than out of season. There are smaller localized outbreaks of different magnitude, and therefore this is more of a seasonal pattern than a pandemic pattern. Of course, the virus is still around, and it'll be our seasonal virus for years to come, maybe 10 or 20 or 30 more years. But that's it. It was a short one, and it's over.
So one of the things I noticed was that during the pandemic, the H1N1 basically displaced the seasonal virus, though I suppose it was out of season. Nevertheless, it was the predominant virus. And now, according to this report, we now have a mixture of the older seasonal strain and the new pandemic, which is now a seasonal strain. And I'm kind of wondering how that happens, where it's displaced to start with, and then the seasonal strain seems to come back, and now we have a mixture. That interests me.
You think migrating birds have anything to do with it?
That's a good possibility that they keep reintroducing them. We really don't know. There are a lot of ideas about how this happens.
I remember when you talked to Peter Palazzi about this, he predicted that the pandemic strain would not completely replace the seasonal strain, and I was sitting here chuckling, thinking, well, he got that one wrong, but actually it turns out he got it right, because now there's a mixture.
Well, you know, some years it does displace. So in 1957, the H2N2 displaced the H1N1. In 1968, the H3N2 displaced the H2N2. But in 1977, H1N1 came in, and it co-circulated with H3N2. And those two viruses circulated until last year. The new H1N1 arose, and now that is co-circulating with H3N2. So the old H1N1 seasonal seems to be gone. And in fact, that's why the vaccine for the fall will be just pandemic H1N1, H3N2, and an influenza B strain. So the mechanisms for this are not clear. Why did it displace for so many years and now not? I don't know. And there are lots of hypotheses, but no one really has a satisfactory answer for that.
So in the end, how bad was this?
Good point.
Was there a significant increase in excess mortality?
Well, that's a good question. CDC has a great surveillance site, and they show, of course, in the U.S., they stopped looking in May, the end of the season, but you can look at the excess mortality. So a good measure for mortality is what's called pneumonia and influenza mortality. And if you look, they have a nice graph of this for 122 U.S. cities from 2005 to 2010. And there was a spike in excess pneumonia and flu mortality in 2010, but it doesn't compare to the spike in 2008, where there was an epidemic, a large epidemic in the U.S. of H1N1, seasonal H1N1. So I would say this did not exceed it. Maybe in certain populations. The pediatric deaths are really higher than they have been. There's another very nice graph at the same CDC site. It's called Number of Influenza-Associated Pediatric Deaths. There's a big spike in 2010 compared to previous years.
Right, which means that in terms of total life years lost, which is the relevant measure for a lot of things, including the economic impact, this was huge.
Yeah.
Because a pediatric death compared to a 65-year-old.
Sure.
But I'm used to thinking of pandemics as causing a huge spike in excess mortality, and that doesn't seem to be the case in this particular case. So I wonder how much our awareness in terms of public health and vaccination impacted on limiting that excess mortality. I mean, is this a big success story?
I think it's hard to know.
Yeah, I just finished working on a flu-related project, which I may discuss or maybe even suggest a guest for in future, depending on how they like the article. But the data on this from the last season, a bunch of organizations collected a lot of data, and we talked about some of that on TWiV. The public health measures, very, very difficult to measure. I mean, what level was compliance? How often did people really wash their hands? What was your control group? So I don't think we'll ever know the answer there.
No.
Not from this pandemic anyway.
Yeah.
The vaccination, the problem there is the supply problems. So the limiting step in getting out the H1N1 vaccine initially was everybody wanted it, nobody could get it. And the virus got ahead of the vaccine. Mm-hmm. And, so that certainly blunted the impact the vaccine might have had. The studies that were done contemporary with that show that it did protect people. So it appears to have had an impact, but it's a little difficult to quantify, was that impact blunted because of the supply problems? And probably.
It also started out weird, too, in Mexico, right? With a lot of deaths, and everyone thought that the mortality rate would be very high. And then that turned out to be from something else. And so, therefore...
Certainly, it wasn't as high as that suggested.
A lot of red herrings in this one.
Right. It came from an unusual place and also had a very unusual season.
Mm-hmm.
Now,
Vincent, this CDC site, FluView, is terrific. That really ought to go in the show notes.
It's a great site.
Yes.
You've used this before, I'm sure, but I've been relatively unaware of it. It's really terrific.
Yeah, they have, of course, now they're not doing any surveillance. They stopped on May 22nd, but they will begin soon.
And that will also go on FluView.
And it goes on FluView, and you can pick what week you want to see. And they have flu-positive tests. You can see a spike in 2010. They have laboratory-confirmed hospitalizations. They have drug resistance, excess mortality, and then pediatric deaths. Really good stuff.
Great stuff.
This must exist for other countries, but I have yet to find it.
Well, the CDC has had a big push lately to put as much of their surveillance data online and publicly accessible and readable as possible.
And they've done a good job.
Yes.
Your tax dollars at work.
Yes, exactly.
It's worth pointing out that in the Southern Hemisphere at the moment, we have the flu season. And there's a nice summary of that at WHO. This is Margaret Chan's summary. Let's see, can I find hers quickly? All right, well, there is a, Anyway, there's a good summary of what's going on there. There are isolated outbreaks in the southern hemisphere. Interestingly, I found that in Africa, for example, it's predominantly H3N2. Here we go. Monitoring patterns and level of worldwide activity. The southern hemisphere season runs from May to September for flu. In most countries, the level of flu reported is low, and countries are reporting a mix of strains comprising pandemic H1N1, H3N2, and influenza B viruses. Where the pandemic H1N1 strain is prevalent, some severe illness and deaths have been reported. Active transmission still persists in the tropics, South and Southeast Asia, Caribbean, Central America, and West Africa. But the kind of details that we have for the CDC here in the U.S. just aren't available. Mm-hmm. So that's a great site. So there you have it, just a little over a year. Pandemic is finished, but we still have to immunize. I'm just curious, how many people got pandemic influenza vaccine last season? I know I did. Did you, Dickson?
No.
Guess not. Michael, did you get vaccinated?
For the H1N1?
Yeah.
I tried, and actually they ran out. Twice.
I almost got it at the Chicago O'Hare Airport, but they didn't have any left.
I actually had the same problem. I tried repeatedly to get. I managed to get it for my daughter because they had pediatric doses available to the pediatrician, but it was simply unavailable to adults until.
Rich, you got it, right?
I got mine. Yeah.
Well,
it's interesting because many places had excess doses,
yes.
Right? So that is some area that needs some help, a little better coordination.
Well, there was because of the timing of the season, it was like the worst possible timing of an outbreak in terms of flu vaccine supply. And the other problem that occurred was as the manufacturers ramped up the H1N1 production, they had to stop the seasonal production. Mm-hmm. So I actually got the seasonal shot very early. The whole family did, and then everybody switched over to the H1N1, but there were a lot of people who never got the seasonal because they couldn't. It evaporated.
Right.
It'd be interesting to know how many people refused the vaccine also from these crazy hearsay stories about the ill effects of vaccination.
Yeah, it's difficult to track. A lot of people just don't bother to get it, and you don't know if they're refusing on principle or if they're just they just assign it a low priority, which a huge number of healthy people do, and even a lot of high-risk people.
Sure.
All right, that's it for the pandemic. Let's move on to another story. And this one concerns dengue in Florida. We've talked about dengue quite a bit.
We have.
And according to, there's a wonderful report at CDC, Morbidity and Mortality Weekly Report. Dengue is the most common vector-borne viral disease in the world. 50 to 100 million infections, 25,000 deaths each year. So essentially, there have been mostly imported dengue in the U.S. Since 1980. Dengue is endemic in Mexico, and they get imported over the border. So it's mainly along the Texas-Mexico border. But in the last part of 2009, a New York physician notified the Florida Health Department of a suspected dengue case in a New York State resident whose only recent travel was to Key West, Florida. Mm-hmm. And so they have several case reports here, case 1, 2, and 3, of various individuals. They all present with fever, headache, chills, malaise, and eventually, after several visits, they were diagnosed as having dengue. These were, again, from people that were in Key West. So what they did was do some surveillance in Key West, and as a result, they identified 24 additional cases during 2009 and more in 2010, and the total bringing up to 2028. So these are dengue, and of course dengue is mosquito-borne. And Dickson, what is the mosquito that...
I was going to ask which one they identified here, because usually it's the Aedes aegypti, but
that's right.
There's also Aedes albopictus, which is the Asian tiger mosquito, which has been implicated in some outbreaks. And so I wondered how the epidemiologists approached identifying
so that
which mosquito.
In response to the dengue in Florida, it's locally acquired in the Keys. They started spraying and, adult mosquito control, but they also tried to find mosquitoes and look in them. So they did some mosquito surveillance.
And they found it, so
407 pools of adult female Aedes aegypti.
Aedes aegypti.
Right.
And they were, 2 pools were tested positive for dengue, so showing that the virus is in them.
So what's the special breeding sites for Aedes aegypti that makes them so difficult to control?
Would that be is this the tree hole mosquito?
It is.
Holes
it's
in
the
trees?
one that drove Walter Reed crazy.
While digging the Panama Canal.
Yeah, because we were creating habitat for the mosquitoes by our own carelessness of discarding old tin cans of food for the workers and things like this. And the mosquitoes, the local mosquitoes, the dengue and the yellow fever bearing mosquito, the same mosquito, by the way, was taking advantage of these new sites to breed in.
Mm-hmm.
So by getting rid of that, you could clear a path for the rest of the construction. So that just knowing which mosquito helps a lot to be able to proceed.
Getting rid of very small pools of water in a large area is very difficult, though.
No kidding. Especially when there's a natural tree hole breeders. I mean, tree hole is not a trivial term. There's a lot of holes drilled in trees by birds like woodpeckers, etc. And that collects water, and when that happens, then to try to control that is difficult.
Right, and every tire track on a muddy road or pretty much any kind of open container out in the open in a rainy area.
Yep. But in a densely populated area, you can do it.
Yes.
It can be done.
So they have a nice summary here. So this is the first dengue cases in the continental U.S. outside of Texas, Mexico since 1945.
Right.
And the first in Florida since 1934.
Right.
And they believe that began maybe around July of 2009.
So how do you think it got to the Key West?
That's a good question.
Yeah, I'm wondering about this too, because if you look at the distribution of dengue fever and look at the distribution of the mosquito,
right?
It's like the mosquito's range extends north, typically, of where dengue fever has been found. There's a sort of a buffer zone there where we have mosquitoes and no dengue, and it pretty much correlates with a U.S. border. So what's going on there?
So what's your guess, Rich?
Actually, I talked to a dengue person recently who suggested that basically cultural differences had an influence.
Air conditioning.
That is air conditioning, right? The people go inside at night. And when it's hot in the U.S., rather than sit out and get bitten by mosquitoes.
No, that's fair enough, but where do you think the dengue mosquitoes caught their dengue from?
People, right?
Yeah, which people?
Yes, someone who brought it in probably, right,
Exactly,
Dickson?
exactly. A visitor from a dengue area came to Key West and gave the virus to the local mosquitoes.
Or somebody from Key West visited a dengue area and then came home.
Right.
All right, but dengue was here at one time.
It was.
Then it went away.
That's right.
Maybe because of air conditioning and screens.
Yep.
But now it's coming back.
It is.
And why is it? Because more travel, more mixing, more...
Perhaps.
All that stuff that is a good...
Or maybe this is just an outbreak, that resulted from an index case somewhere in Key West, and it'll go away and we won't see it again for 50 years.
Except if this is a vertically transmitted virus, which I believe it is, because all the flaviviruses are vertically transmitted in mosquitoes, you can maintain this in the population without having to bite anybody.
Mm-hmm.
Okay.
So that's, it maybe establishes and it'll take a longer time to go away as the virus gets diluted out by bad weather and perhaps not high temperatures allowing higher viral titers to develop inside the mosquitoes. Maybe that'll burn it out, but the mosquitoes can harbor it for quite a while without having to bite people.
So Dickson, what's the range of Aedes aegypti in the U.S.?
It's limited. It is limited, and we keep track of that mosquito because of our long history with yellow fever in this country. In fact, it nearly decimated, and I mean 1 in 10 in this case, the population of New York City back in the turn of the century. We had a huge outbreak of yellow fever, right? And we cured it by going to church, as I recall. No kidding. They actually said, go to church and something will happen. And because everybody went indoors and stayed indoors for quite a while, it actually broke the transmission cycle for yellow fever, of all things.
So since yellow fever was a problem,
Yeah.
I mean, between then and now,
Yeah.
have human control measures restricted the Aedes aegypti vector?
It has. And along, in densely populated urban areas, it's easy to identify these breeding sites and to get rid of them. Which is what Walter Reed did, and which was what a whole bunch of other people did to keep the vectors from actually biting people. They'll bite other things too. They'll bite monkeys, right? That's the way they maintain yellow fever down in South America, not in people, but in monkeys.
Yeah, one of the things I discovered in sort of reading up on dengue in preparation for this is that it is maybe one of the only or the few arboviruses that can maintain a cycle just with humans in urban areas, whereas some others
you're right.
need the sylvan, that is wilderness cycle as well.
Yeah, we're the only host for dengue virus.
I think there is a monkey host.
There is a monkey host?
But it's not essential.
Okay.
Right.
It doesn't play a role in the transmission cycle.
Well, in certain areas it may, but not here. Yeah.
Okay.
This
it can help maintain a reservoir, but it's not required as for some other arboviruses.
I'm glad I was able to reveal my ignorance once again.
Well, we can cut it out, Dickson.
No, leave it in. I love the hate mail I get.
Someday we're going to be live, Dickson.
This is true, and then I'll really, brother.
So there's another mosquito host, right, Dickson?
Yeah. Aedes albopictus,
and that's more broadly distributed.
It's the Asian tiger mosquito. We imported it into this country. We've had a prior episode of dengue in which we discussed the vectors, and this was a vector brought in through retreading U.S. tires in Japan, where the Asian tiger mosquito was endemic. They actually breed inside the tires that are laying dormant, waiting to get retreaded, but they accumulate water every time it rains, right? So they lay their eggs along the water line and wait for the water to go down and then go back up again. When that happens, of course, bingo, you get more mosquitoes. The tires were imported back from Japan without water in them, but with the eggs. And they were stacked up outside the factories, I guess, for re-rimming or whatever they do with them after they retread them. It rains, it activates these mosquito eggs, and they start to fly away. And they know exactly where they came from. Somewhere in the Midwest, maybe Indiana was the epicenter for the introduction of the Asian tiger mosquito. But what they've learned is that this mosquito will outcompete Aedes aegypti for breeding sites. And it's not as good a vector for yellow fever. I'm not sure if it's as good a vector for dengue or not, but I know it's not as good a vector for yellow fever as Aedes aegypti is. So it's a blessing in disguise. Maybe there's some fear of introducing yellow fever back into the United States, and the Asian tiger mosquito standing there says, "You may not come in." So it's a very interesting interplay between invading new species and old species of mosquitoes and how they behave next to each other.
The secondary vector, albopictus, has spread throughout the southeastern U.S. and was responsible for a dengue outbreak in Hawaii in 2001, likely after the virus was introduced by a Hawaii resident returning from Tahiti. So there you go. I don't know. I think this is going to spread. That's my prediction.
Yeah, I would agree with you there. I would. Definitely.
There's a great page on dengue at the CDC, which we'll put a link to. And in particular, there's a page on epidemiology, which summarizes transmission. They give a nice history. Did you know, Dickson, that the viruses originated in monkeys, the 4 serotypes, and jumped to humans in Africa or Southeast Asia between 100 and 800 years ago? Was a minor disease until the 20th century. Disruption of World War II. Transport in cargo of the mosquitoes are thought to have played a crucial role in dissemination. And then it was documented first in 1950s in Philippines and Thailand, and in 1981, large cases in the Caribbean and Latin America.
Interesting.
It spread quite a bit. For a while, it was limited because we did a lot of mosquito control, right?
Right.
With DDT.
For malaria, for a totally different reason.
Yeah.
That's right.
And we stopped doing that, correct?
Exactly.
So now it's back.
This MMWR report, by the way, is really good reading. It's easy to read. We've talked about this before, how good these reports are. I find these case reports absolutely fascinating.
Good stuff.
And then the editorial notes on the bottom that are, what was good and bad about this. And they particularly laud, although it took a while to figure out what was going on in the index case number 1. It didn't take too long, and they talk in the editorial about how important it was that diagnosis was done in a timely fashion, because that clued them into what was going on in Key West. And it's particularly interesting that was a person in Rochester who'd been to Key West. And so it took them a while to put the whole thing together. Very interesting.
It's an interesting graph at the end showing the onset of the cases, or the number of cases as they were picked up either by case reports from physicians, medical record reviews. They went back and looked at some records, and then household serosurveys. And those are the 28 cases. The distribution is very interesting. 19 in males, 9 in females. Mostly within the 21 to 60 age group. 24 white, 3 black, 1 Asian Pacific Islander.
Now, did I understand from this? Okay, so lots of times dengue infection doesn't cause any symptoms, right? And my reading tells me that this is actually quite variable depending on the circumstance.
Right.
The ratio of non-symptomatic to symptomatic symptoms can be quite variable. However, in some cases, there can be as many as 90% of the infections that are asymptomatic. So there could be quite a few more infections out there in Key West that have not been identified. But they did pick up in their serosurveys that this count that they have includes both symptomatic and non-symptomatic people. Is that correct?
They were picked up on serosurvey. 2, 4, 6, 9, 10, 11 were just on serosurveys. So we don't even know if they were symptomatic or not.
Right.
And we should say that the first infection is typically not terrible with dengue, but if you get reinfected with a second serotype, then you can have the very serious shock syndrome, and that
which is what it's known as breakbone fever.
Breakbone fever,
We've had several of our medical students, by the way, who went for medical electives in the tropics, which I used to oversee here, and come back. We had 2 of them actually admitted to the hospital with dengue. Yep.
So it says here, all the cases in the U.S. have been imported.
That's right.
This is really the first in a long time where the infections seem to have happened right here.
Exactly.
Very interesting. All right. The next story is Vaccinia virus infection in monkeys in the Brazilian Amazon. And this was originally a paper in Emerging Infectious Diseases, and in the same issue is a commentary by none other than Rich Condit.
Hey, I know him.
How about that?
So he must know something about this.
I hope so.
Why am I laughing? I don't know why I'm laughing.
You're just a funny guy, Dickson.
Don't pay attention to me.
Okay.
I like the name of your article, Whence Feral Vaccinia.
Yeah, I had to look up whence to make sure that I was using the word correctly. It came into my head, and I wondered, am I really using this right? But it turns out to be okay. It's like, where did this come from? So I want to remind people that we're talking about pox viruses here.
Right.
And the most notorious of those was, I can use the past tense for the most part, smallpox, which was a really nasty disease that was endemic and epidemic throughout the civilized parts of the world for centuries until it was declared eradicated due to vaccination with Vaccinia, the smallpox vaccine. The eradication was declared in 1970, well, depending on who's counting, 77, 78, 79, and is the only virus disease to have actually been apparently cleanly eradicated. It's been 30 years now, and we've not seen another naturally occurring case of smallpox. The disease is characterized by this really nasty pus-filled blisters all over the body, and usually they're about 30% case fatality rate from septic shock. And the vaccine that was used was a virus that was isolated from the wild, from animals from cattle that had a disease called cowpox, which was a much more limited blistering infection. And in fact, what was discovered is that you could take some material from blisters from these cows and artificially introduce it into the skin of people, and they would get basically a single blistering lesion that would confer protection to smallpox. And this was known because people who milked cows got this disease accidentally, and they were apparently resistant to smallpox. So that virus is called Vaccinia virus. It's very closely related to smallpox, originally came from nature, very closely related to smallpox, but causes a limited pathogenesis, enough serocross-reactivity to immunize somebody against smallpox. Interestingly, if you look at what we call cowpox today, look at the genome or whatever, it's different than Vaccinia, and it's not clear where what we call Vaccinia really came from. Whether it was a virus that was present at the time of Jenner, who was the physician who did this stuff 200 years ago and has since disappeared and has been maintained only in laboratories, or whether it evolved in the hands of humans during the various vaccination programs, it's just not clear. But in most cases, you don't find this virus in nature, with a couple of exceptions. One is buffalo pox in India. This is a disease, obviously an exanthemus disease of buffaloes that's caused by a pox virus. I'm not entirely clear on this, whether all buffalo pox is Vaccinia, but at least some of it, if you isolate the virus and look at it, is in fact Vaccinia virus.
So it's the vaccine strain, clearly.
Well, it is Vaccinia virus. I don't know if in that case, I'm not as up to date on that one, whether it's clearly established that it is the vaccine strain that has been reintroduced into the wild.
Okay.
But in the case of Brazil, the evidence, in my mind at least, is pretty compelling. So over the last 10 years, first reported in the year 2000, in fact, by my friend Nassim Musashi, there has been a reemergence of cowpox disease in Brazil. It shows up in cattle and is transmitted to humans, and it's although it's not a really devastating disease, it's certainly a nuisance. And that was identified as a pox virus, and furthermore identified as Vaccinia. And furthermore, on a closer inspection, it turns out that in a particular gene in this virus in the wild, there is a signature deletion that Nassim had the foresight to look at the virus that was used in the smallpox eradication campaign in Brazil, which is they had their own virus strain, which is fairly unique, and it contains the same deletion. Okay, so there's reasonably compelling evidence that in this case, in the stuff circulating in Brazil, it's the Brazilian vaccine strain that has established itself, at some point, in nature, and was probably pretty much kept quiet by the ongoing smallpox vaccination campaign. But now that we're no longer vaccinating against smallpox, since it's been eradicated, there is now a pool of hosts, susceptible hosts that this thing can reemerge on. And I think what we're seeing is reemergence of the vaccine virus that had been reintroduced into the wild. Very interesting.
What's the consequence for the monkeys?
So this paper asks the question, what is the reservoir for this virus in nature? And the authors assayed a bunch of animals that had been collected during a project where a hydroelectric dam was built in the state of Tocantins, if I'm butchering that pronunciation, in Brazil, which is kind of on the edge of civilization south of the Amazon forest. And when they created this hydroelectric dam, they collected a bunch of animals to relocate them. And this made available, and they, of course, did a bunch of veterinary shtick to the animals, including collecting serum and stuff. So this made it available to the investigators, a bunch of samples from these animals who had very limited, if any, contact with humans, okay? And it included a wide variety of animals, and they tested them all for Vaccinia antibodies using a plaque reduction neutralization test. You take virus, you react it with serum, and ask whether the serum will neutralize the virus based on whether or not the virus can cause plaques. And they also did PCR to see if they could find viral DNA. Yeah, I guess that was about it. And what they found was, let me see.
What kind of animals? Just monkeys?
They had I gotta look at that. They had mostly monkeys, but they had, let's see, here it is, 269. How do you pronounce this? Capuchin monkeys?
I always said Capuchin, but I don't know if that's
Capuchin,
right.
you're probably right. 27 black howling monkeys, 12 coatis.
Coatis?
Yeah.
What are they? I looked this up at one point.
Yeah, they're cute little
raccoon-like animals.
Raccoon-like animals.
Long tails.
I learned this from a children's wildlife show.
Right. 20 agoutis.
Oh, yes.
Yeah.
2 possums. 5 armadillos.
Them's good eating.
And a partridge in a pear tree.
5 collared anteaters and 4 gray foxes.
Cool.
Wow.
Okay. I think they had a couple of extraneous positives in some of the other animals, but mostly, let me see, 84% of the animals, predominantly monkeys, were seropositive for orthopox virus. That's 24%.
Did that include the possums, or were they just faking it?
Good, Alan. I don't know that I'd have to look here again. I don't know that any of the possums were.
It was not a serious question.
I know, but let's get the data. Let's seek the truth here.
Here we go. It's in this table here.
68 of the Capuchin monkeys were positive.
Right.
27 of another species of monkey were positive.
Basically,
two coatis, 1 agouti. Sorry, no possums.
Yeah.
So if they were faking it, they were good at it.
Yeah.
So mostly monkeys. 24% total of the animals were seropositive. That's a big number. All right.
Yes.
And it
is.
Says that 18 of the serum samples were positive for orthopox virus DNA. And when they did some sequencing on some of these samples, and sure enough, the virus has this signature deletion. It looks like the virus that's circulating elsewhere as a sort of a cowpox-like disease. It's the same stuff that looks like the vaccine strain.
Well, not only is the prevalence pretty high among these animals, but the diversity of the species they surveyed is incredible.
Yes.
Rich, did they sequence every isolate? So do we know if they are somewhat different? That would indicate that they're all independent isolates?
No, the sequencing is pretty limited. I don't think there's enough sequence information to know whether they're identical or whatever. And they only sequenced 6 isolates, and they just did parts of 2 genes. Okay, so I don't think there's enough sequence data to, build a really elaborate tree or anything like that.
So how did it get? The idea is it went from people to these animals in some way, right?
Yeah.
How'd that happen?
Well, okay, so this is anecdotal. Who knows? I don't know. But I have contacts in Brazil who are still working on this problem. And I email them back and forth occasionally. And in one exchange a couple of months ago, the story was related to me that in digging around in this, it came up that at some point during the smallpox eradication campaign, one thing that was done was I believe it was to actually vaccinate cattle and give them to farmers as a reservoir for vaccine. Okay. So as if the vaccine was being propagated on animals in rural areas for the purpose of making vaccine available. So there's a way that it could get back into the wild. But it is quite possibly, if you just vaccinate people and you get a few accidental vaccinations and they're out in a rural area and they're farmers and they're messing around with farm animals, you could get some limited transmission to the farm animals.
Well, people that live in the bush in those regions, they live with their animals very closely.
Yeah.
They live with their pigs and dogs and cats. That's why Chagas disease is so prevalent in those regions as well. Plus, this is the most biodiverse region in the world for mammal species by far. This has many more mammal species than any other place on Earth because they have very few predator species that live there.
But now one of the key things here is we have apparently a really high incidence of this virus out in an area where there's been very little human contact. And I guess very little farming, though if you look at the satellite picture, there's quite a few farms.
Right. Well, they built a hydro dam there, so there must have been a lot of workers there at one time too, right?
Yeah. So I really, it's remarkable. And it suggests, though doesn't state, that there's a very significant reservoir for this virus in animals, predominantly monkeys in the wild. One of the questions I had, I'm not used to being able to get Vaccinia virus DNA out of animals without the animals being symptomatic. Mm-hmm. Okay, this virus is there's very few asymptomatic infections in my knowledge. So I was wondering whether there was in the veterinary reports anywhere any sort of information on whether these animals had any disease, and I couldn't get any satisfactory answer to that question. I want to see a completely independent study on this, another lab go out and do exactly the same thing, maybe in a different area, and get the same result. If it's confirmed, it's really a remarkable finding.
Is Brazil the only place where we've found this now?
Well, there's the buffalo pox in India.
India, that's it.
Right, and there's the issue of where have we looked.
Yeah,
right, exactly.
Yeah.
But this also
I think what needs to happen is other groups, preferably in very remote areas, need to go out and start sampling animals and see if you find this stuff.
Right.
I was just at a seminar today. A woman from Croatia who works mainly on hantaviruses. She isolates them from wild mice in various parts of the country. And she showed one slide where they had also isolated pox viruses from these mice.
I'll be darned.
A high frequency of isolation. So I asked her, what kind of pox viruses are these? And she said it was only a sero survey, so they couldn't identify whether it's Vaccinia or monkeypox or something else, right? But it would be interesting.
Now, was this study done in Croatia?
Yeah, it was.
Interesting.
So
I'd
that
like
would
to
be
know
very
what
interesting, right? Because if it's Vaccinia-like, that means it's in more than one place. Yeah.
But there's a lot of these viruses around, and rodents are the most typical reservoir for these. That's where they're most typically found. I would not be at all surprised in this particular case if you're talking about cowpox, more classic cowpox disease.
Right.
But who knows?
Could be. No. She said it could be cowpox, it could be Vaccinia, or I guess monkeypox, but probably not, right?
Probably not.
You don't get that out of mice, yeah.
When we developed the vaccine, of course, did we know that this virus was that promiscuous?
No, not really. I mean, we knew that it grew on cattle and people, and that was it.
Exactly.
And it wasn't recognized until, it wasn't recognized until much later that Vaccinia actually grows. Vaccinia itself has a fairly broad host range. It'll infect quite a variety of animals. As I said, most typically the host in nature for these orthopox viruses is rodents. And by the way, Vincent, that does include monkeypox. Rodents are probably monkeypox is a misnomer. Rodents are probably the natural reservoir, and the monkeys pick it up by eating or messing with the rodents, and the people pick it up from the monkeys.
Right.
Cool.
Very nice story, Rich.
Yeah, really interesting
So
story.
when you say a pox upon your house, you're not actually sure which one is this one.
No. And if you're really a nice guy, it's a Vaccinia upon your
It's a
house.
cowpox on your house.
Yeah,
That's the
exactly.
nicest thing you could say to anybody.
That's right.
All right, our last story is a paper that actually was a pick of the week at ASV TWiV by Marilyn Rusink, and it's called Viruses in the Fecal Microbiota of Monozygotic Twins and Their Mothers.
And it's published in Nature, so that's FAECAL.
Indeed.
Right.
That's right. Yes, it's roots. This is an amazing paper. I thought we should go into this a bit because Marilyn just really mentioned it. as a pick and didn't talk about it at all. So what was done here was to get fecal samples from 4 pairs of adult female monozygotic twins and their mothers. Monozygotic from one egg, okay? And the point was to see, is there any difference in if you're a twin, or is there any difference between you and your mother? So they got fecal samples at different times, the beginning 2 months later and 1 year later, and that was to see if there's any variation. And they purified virus-like particles from these fecal specimens. So they did not just deep sequence the nucleic acid from the feces, but they purified particles. And then they sequenced the particles. Okay? They got 96 million bases of sequence. So the bioinformatics in this paper is amazing. What I've just told you, getting all these fecal samples is amazing too, and purifying particles. 96 million bases, and 4,193 genomic viral sequences were obtained from these 32 specimens. 73% were eukaryotic viral genomes, and I'll tell you what viruses in a moment. 25% were phages, viruses of bacteria. And 77% of these phages were mostly double-stranded DNA phages that infect bacteria that are resident in your gut.
Right.
Proteobacteria firmicutes. Does anyone know how to say that?
Mm-mm.
Firmicutes.
Firmicutes,
and actinobacteria. Amazing. Now here's the cool part. 81% of the sequences did not match any known viruses.
Really?
81%.
Yeah.
Brand new. And most of the phages were temperate phages, which means they don't kill the bacteria, but they stick their genomes into them. Right, so those are some of the numbers. Let me tell you a bit. Now,
but
this
did
paper.
The twins share common biota?
That's a good question.
That was the point of the paper, I hope.
Now, just
a
brief bit of a side note here. Before this study, going back just a few years, others have done bacterial surveys of the intestine.
Right.
And what you find is families often show strong similarities in the bacterial complement in their guts.
Right.
So you would expect,
not
based
a surprise.
they're exposed to similar things. They're eating meals together. But you find an enormous bacterial community.
Yes.
In the gut, and you find that, say, a mother and 2 twins would be very likely to have nearly identical bacteriomes within their intestines.
Yes, good point. In fact,
back to the viruses.
In this study, they actually also did some sequencing to identify the bacterial communities as a control.
Sure.
And what you do is you sequence the ribosomal RNA of the bacteria, and you can very quickly type what kinds of bacteria are present. And there are about 800 bacteria species in us.
We know so little about our intestinal tract. It's incredible.
We're getting to know more, Dickson.
We are.
Now, this paper focuses actually on the phages, but I just want to, and they say very little about the eukaryotic viruses, which were really the highest percentage of all their sequence. So I guess we'll see that in another paper. But they do say what some of these viruses are, and this is very interesting. First of all, they didn't get more than half of any genome of a eukaryotic virus, so it was hard for them to sort out what's going on. So they got 6 non-human herpes viruses. Non-human.
So, is this in the supplementary?
Yeah.
Stuff?
Yeah, it's in the supplementary data. So 6 non-human herpes viruses, 1 plant virus called grapevine fleck virus. So I guess they ate some grapes. This came through. 1 reovirus, an aqua reovirus, 1 parapox virus, bovine papular stomatitis virus.
Excellent.
Now tell us about that again. This came up in another TWiV, right?
Yeah, it's just a different. It's a fringe pox virus.
What does it infect?
Cows.
Cows.
It can infect people.
So maybe this person just ate some steak and this is coming through.
These kids are only a year old when they took these samples? I mean, when did they detect them?
They were older, weren't they?
These are older, yeah.
I mean, in the beginning, they shouldn't have detected anything except what they got from their mother.
Yeah, I don't know what the twins, but they are not just babies. They're older, yeah. Maybe someone can find that while I'm jabbing. And one rotavirus. So maybe someone had a rotavirus infection. So not a lot of detail on the eukaryotic viruses, but there are a lot there, and maybe we'll hear more.
Non-human herpes viruses. Is that what you said?
Yeah, non-human. That's all they tell us.
Where is this in the supplementary information? I've got it in front of me here, but I can't.
Page 3.
Page 3. Okay.
I don't know which non-human herpes viruses. You know, there are reptile herpes viruses, amphibian herpes viruses, fish. Maybe they ate fish.
Reptiles.
Reptiles. So anyway,
Burmese pythons.
I mean, this could be stuff that's passing through, right? So
right.
Anyway, they focused on the phages in this paper, and the story is just remarkable. So first, Dickson's question. What is the percentage of shared viral sequences between samples obtained at different times from the same individual and from different individuals, right?
Right.
That's what you want to know.
That was basically my question. Thank you for formalizing it.
The same individual harbors very similar fecal viral communities over a 1-year period.
Mm-hmm.
Greater than 95% of the genotypes were present with minimal variation over the year. Okay, and they say this is different from other comparably characterized ecosystems where almost daily changes in diversity occurs. So it's a very stable population. So minimal variation within one person over a year, but between people, very high variation. So the interpersonal variation in the phage communities is very high.
So this is why only your own smells good.
One way to put that, Alan. That was very poetical.
And some don't smell at all, right?
That's right, sure.
So what they find is very interesting. You have your own unique population of viruses, but there are always a few very high abundance phages, and that those few differ from person to person. Okay, so and they compared this to the bacterial communities, which, as Alan summarized before in other studies, they're quite unique, right, depending on what you eat.
Right, but mainly within the family.
Within the family, they are stable. But the viral species are quite unique.
So does that mean that you get your bacteria from your intimate contacts, but you get your phage from elsewhere? Or does it mean something more about how the phage population is established in your gut once you contract them?
Keep in mind, these are infants, and they get a lot of diarrheal disease, so you get a lot of turnover for microbes.
I don't know if they're infants.
I don't think so. 2-month to 1-year-old kids. That's what I gathered from what you said.
No, I don't think so. They took the samples at year 0 and 2, or at time 0.
Time 0. I'm sorry.
Time 0, and then 2 months and a year later. But
okay. I thought that was the age of the people.
The subjects were actually older.
I'm sorry about that.
Yeah,
that doesn't rule out the possibility.
No, they seem to favor the latter, Rich, that there's some selection that goes on and that gives you a stable population of phages.
Right. So you may be exposed to the same spectrum of phages, but once exposed, the dynamics can be different, so that the stable population that you establish can differ from individual to individual.
And I think there may be the establishment of the virome may be subject to more variation from individual to individual because the bacteria have to be there first, right?
Well, they can bring in lysogenic phages with them.
They could, right? So, but you've got the bacteria coming in and establishing early in life, establishing their ecosystem within your gut, and you have the viruses coming in and establishing their ecosystem within the gut. It seems like the viruses might have more, I don't know, flexibility to establish different viromes within a family.
I don't know why everyone is different then. You know, you would think there are common things that we all need.
But why questions in biology are
can't answer them.
Impossible to answer, not just hard.
The next thing they did, and they said, what about the proteins encoded by these viral genomes? Can we learn anything from them? So they looked at all the open reading frames of these viral genomes and compared them to the open reading frames in the bacterial genomes that are resident. Okay, so the viromes versus the microbiomes. And they have this wonderful figure that breaks up the proteins into different classes, replication and repair, amino acid metabolism, carbohydrate metabolism, both for viruses and bacteria. So they're broken down according to function. And what you find is that there is very little interpersonal variation in the proteins that you see in the microbes in your gut. You can just look at the colors, and you can see among all the samples, the colors are very much the same. But among the viral genomes, each individual has a different complement of specific proteins that do different things, involved in energy metabolism, lipid metabolism, transcription, signaling, sugar metabolism. It's really amazing. So the bacteria are quite stable, but the viruses are very different. And so these proteins must be conferring some property to the bacteria of some sort. And it's very host specific. I find that really amazing.
Yeah, it's really interesting. By the way, I got the answer to the age.
What's the age? Yeah.
I just lost it again. So it's Missouri adolescent female twin study. That's where they got their samples.
Okay.
Composed of female twins pairs born in the state of Missouri between 1975 and 1986 and their mothers. So the twins are 25 to 35 years old.
Got it.
So there's a lot more in this paper, but I want to wrap it up with one more thought. And this is a very interesting paragraph here. They say, in most ecosystems where phage-host interactions have been studied in detail, lytic life cycles and red queen dynamics seem to dominate. Okay, so in other words, in all the bacterial virus systems that they've studied, like bacteria in sludge, in acid mines, the viruses kill the bacteria, the viruses become resistant by evolution, and then the viruses evolve to overcome those defenses. Okay, so for example, they say the clearest example of red queen dynamics between phages and their hosts are the CRISPR systems, the CRISPR elements. Virus infects a bacteria, the bacteria keeps a piece of the viral genome, uses it to be resistant to the next round of infection, and then the phages mutate to overcome the CRISPR. So that's why it's red queen, because you keep running.
It takes all the running you can do to stay in one place.
Yes, exactly. And they say, okay, that's in most of the other systems, but they found no evidence of this type of behavior in fecal phages. And fecal, F-A-E-C-A-L, phages, P-H-A-G-E-S. They said, we found high abundance of dominant phages present in the same individual for extended periods of time with no significant divergence or mutations in their genomes. This is consistent with the notion that they represent prominent temperate phages in the fecal microbiota. So it's very different from what happens, say, in the ocean, where you have 10 to the 21 infections per second of bacteria with lytic viruses. Here it's very stable. It's mostly lysogenic, and maybe the genes involved are contributing something to the bacteria and to the host. So it's an enormous amount of fodder for future studies here. This is really amazing. No pun intended. That's right.
So you remember I got my degree at Notre Dame where I worked on germ-free animals.
That's what this figure is.
And
I wasn't going to go into it.
No, that's okay, because I mean this is something that's gone around and come around now, because in the very beginning of the work on germ-free animals, people doubted the validity of it, and they didn't actually know why they were doing it, other than just to say they were disproving Louis Pasteur that said life can't exist without microbes. And in this case, it's a tabula rasa for the way we start our lives, but not how we finish them. So when you're born, you have no microbes. And very, very soon after that, you've got a very stable unit of microbial life living in your gut tract, which at the bacterial level looks immutable, but at the viral level looks totally permeable and permutationable. That's not a good word, but that's, what I meant.
Yeah, you were a pioneer, Dickson.
So that's the... No, I wasn't working... Well, you know what I did. I wanted to know whether worm infections in animals could exist apart from microbes, and the answer is yes and no, because they were quite variable. But this is a beginning of finding out who we are. Who are we? And we are this.
Dickson, there was a talk at ASV where so you infect mice with polio, they get infected, right? If you take the bacteria away from the mice, they don't get polio.
Neat.
Example.
It's all connected, kids.
So anyway, Dickson, they had a hypothesis in this paper. They say that what probably happens in the gut is there's energy limitation in the feces. There's not a lot of nutrients there.
Right.
So the phages come out and specific viruses predominate. So they used gnotobiotic mice to address that.
Okay.
One of the authors of this paper, the lab head is Jeff Gordon, but one of the authors is a fellow named Forrest Rower. Does that ring a bell with anyone?
No.
So Forrest Rower is a co-author of a book that Grant McFadden picked as his pick of the week. Coral Reefs in the Microbial Seas.
Okay.
Look at that.
So Mary Ewell suggested we have Forrest on TWiV. She said he'd probably love to do it. So we're going to get him on.
Vince, let me just interject here another thought too, and that is that when you write a grant and you direct it towards the National Institutes of Health, there always has to be some health connection.
Okay.
Try writing this grant and getting funded for that kind of work, because the health connection here hasn't come out yet, but I'm certain that there's a big one. Absolutely certain. The more you find out what normal is, the more you can compare it to the abnormal.
Yeah.
So no one supports research to find out what normal is. And this is finding out who we are. For the very first time, where you're employing high technology to the infrastructure of our DNA, RNA, and all the other living entities that happen to take advantage of us as a successful system. There's a big philosophical component in this thing that isn't stated right away, but it's obvious by looking at it, what it implies. That is, every single person is different, and yet every single person is similar.
Dickson, this is supported by NIH.
It's amazing. Maybe there's an enlightened study section out there that actually senses the need for this.
So there's a study section that can actually tell this from Shinola.
Exactly.
So I have one more question about this paper. This is focused on monozygotic twins, and an appropriate control would be fraternal twins.
Absolutely.
I would assume that you get the same result with fraternal twins, because this is, I can't imagine that the genomics of the people really has much, if any, of an influence on this. It's more their environment. What do you think of that?
Well, the controls here are the mothers, right?
Yeah, I suppose so.
Yeah, but that's similar genetics.
So. I mean, they're all different.
Adopted twins at birth. You know, if you had identical and fraternal twins that are adopted by different mothers, that's your other control.
Right, because so genetics are the same, environment is not.
Exactly right.
Adopted at birth would be difficult, though.
Not necessarily in some places, but
right, in some places.
Oh, you're right. It's a difficult. Twin studies are always difficult anyway.
Yes.
And the conclusions based on them are even more difficult.
And twins adopted to different families at birth would be
hard to
find. But if you could find them, they would be incredibly valuable to medicine. Right?
Absolutely. But there's a lot of twin studies out there. Those people are known for other reasons. And
sure.
Fascinating altogether.
It's a great study, isn't it?
Yeah.
Wonderful.
Yeah. Raises a lot of
So I thought we should give it justice, because it's really fabulous.
Way to go, Vince.
There's even more to it, but that's all time we have. Let's do a couple of email. The first one is from Jim, who writes, some listeners might benefit from reading The Treatment. Why is it so difficult to develop drugs for cancer? In the May 17th issue of The New Yorker, he provides a link. One part of the article describes the mass screening of chemicals for potential compounds and points out that although industrial research labs like Kodak and Fujifilm have millions of chemicals in their vaults, Pfizer was screening only 600,000 and Merck about 500,000. One researcher obtained a grant and purchased 22,000 chemicals from Russia and Ukraine at $10 each. then screen them 96 at a time and 100 batches a day with automatic equipment to find some suitable candidates for clinical tests that eventually produced L-esclamol. They never explained why Kodak and Fujifilm were not approached. Has anyone made an effort to locate where such chemicals are assembled and how they might be accessed? Anyone have insight in chemical libraries?
Yes, this is part of my regular beat in pharma and biomedical research. The process that they're discussing here is high throughput screening, which is what the pharma industry hit upon about 10 years ago, maybe 15 years ago, as the solution to kind of running out of ideas. You weren't finding a whole lot of new drugs in nature, so let's see how many compounds we can synthesize and then screen them to see if they're good drugs. And this approach has been criticized, and it's also been pointed to as possibly the only solution, and people are a little bit polarized about it. But the process starts with synthesizing a whole bunch of compounds. It's normally done by a technique called combinatorial chemistry, where you sort of mix and match different building blocks. And then you move to a preliminary assay where you're going to screen thousands of compounds per day or sometimes per hour against some very, very simple, easy to read readout. Does this stop tumor cells from growing in a well of a 300 and some odd well plate? Or a 1426 well plate is now, I think, the new standard. So they're up to thousands of wells per plate, or 1,000 wells or more per plate, and they're doing the simplest possible assay at the outset, and then subsequent screens on the compounds that meet that criterion would try and narrow things down. Well, is this a good drug? So in terms of why you might not approach companies like Kodak and Fuji, yes, they have millions of chemicals in their vaults, but part of the problem for drug companies is they don't just need chemicals, they need chemicals that could at least in principle be non-toxic, efficacious drugs.
Right.
And that narrows things down considerably. There are some whole classes of chemicals that you wouldn't even want to really consider. And so there's a huge focus in pharma. They've been doing this for more than a decade now, and as we all know, pharma companies have not been producing huge quantities of new, highly effective drugs. They still kind of trickle out. So there's a huge focus now on trying to improve the libraries and not so much screen more compounds, but to screen better ones. This Russian and Ukraine library he talks about, this is actually, there's at least one company that markets that library now. And these chemical compounds come from universities in Russia, where apparently a requirement of certain levels of chemistry degree is that you synthesize a new compound. New meaning new to science.
Wow.
So, to get your chemistry degree, all right, make something.
And what a great idea.
And as a result, these institutions have these huge, tens of thousands of compounds that their students have synthesized in moderate quantities, and they're just sitting around. And so this researcher, and as I say, I think there's a company now marketing this as a general purpose library. I got a hold of them and screened them against some assay, and yes, you can eventually get maybe a drug out of that.
So the drug he's talking about, L-esclamol, is being sold by GlaxoSmithKline as a drug that triggers apoptosis in cancer cells.
Yeah.
So chemotherapy adjuvant.
Interesting.
So that was synthesized by a Russian chemistry student.
Most likely. It might have been faculty, but most of those compounds, my understanding is that they were thesis projects.
Well, of course, this could have been a derivative of the original hit.
Yes.
Right.
That's the other thing.
Most
You do
likely,
a process called lead optimization, where you take your original hit and you start tinkering with it and redesigning it and working on things like toxicology and pharmacokinetics and trying to improve its profile.
So, Alan, would it be safe to say that the Kodak and Fujifilm chemical libraries might not be appropriate for these applications?
I wouldn't rule out the possibility that there's something useful in there.
Yeah.
The problem would be finding it.
Yeah.
Well, let me just add this. Back in the old days when I was a technician here, back in 1962, there was a drug that Kodak produced called dithiazanine. Dithiazanine, a photochemical of some sort that was tried out in mice, shown to be non-toxic, at least at some level, and it turned out to be a cure for strongyloidiasis, which is a very dangerous worm infection. And they gave out millions of doses of this drug until somebody died because they had malabsorption syndrome. And instead of killing the worm in the gut, it actually absorbed across the gut tract and killed the patient. And that's when they took it off the market, and they learned their lesson, and they never made the same mistake twice, obviously. So I think Kodak Chemicals has been off people's lists ever since as go try those next. We don't miss them in that sense.
You can buy libraries of chemicals as well.
That's
is
right.
Quite a big market, and they are often very specifically designed. So if you're looking for particular classes, you can buy libraries.
Chemical and Engineering News have advertising pages filled with permutations on a theme, so to speak. Sure. You can see them all the time, and they're all for sale.
And there are also libraries that you can buy that are a smaller subset. You can buy a library of all approved drugs.
Yeah.
Yeah.
Which is usually that's actually where most people will start when they're trying to work on something for a brand new indication.
Good idea.
Is you go to the library of approved drugs, because if your answer is in there, you're all set.
Right.
Yeah.
You know, all you've got to do is now you know it's safe, you know it's absorbed, you know it's pharmacokinetics, and you would need to set up a clinical trial for your new indication, but your 90% of your work is done.
Right.
So Alan, since you spent some time studying this, can you compare the sort of efficacy of this sort of high throughput screening approach to the more targeted designer drug approach that people talk about, at least?
They're kind of similar in that both have been touted as the salvation of the pharma industry, and neither has quite panned out as such. Mm-hmm. So rational drug design sounds like a great idea. Let's start with the science. Let's design a drug that targets things very, very specifically. And, it gets us around all this, what some people would call brain dead screening. And yet some of the compounds that have come out of that, and sometimes they don't work out so well.
Right.
Mm-hmm. A lot of them have turned out to have kind of middling or poor efficacy and toxicities they didn't anticipate. And the reason for that is not that the drug designers were not thinking, it's that they didn't have enough data.
Yeah.
Because we don't know everything. Right. And that's the fundamental problem we're confronting.
We do know what the pocket of the enzyme looks like, however.
Oh, sure. We know
but that doesn't answer all the questions.
You can design a drug that fits into a particular three-dimensional structure found by X-ray crystallography, but that doesn't tell you all the forms that protein assumes. It doesn't tell you everything else it could bind in the body. And so neither approach has been as helpful as people had hoped. However, both have produced successes.
Right, so you got to basically keep both alive.
Yeah, and the other thing is, you got any better ideas?
Right.
Avoidance. So those are the two main approches, and I think
don't get sick.
Exactly, don't get sick is the solution.
Yeah, that's the
best.
Avoidance. That's right.
That, by the way, was Jim from Virginia. He sent a link to the article. It's behind a paywall, so what you get is the article, but it's too fuzzy to read. But it is written by Malcolm Gladwell.
Oh, yes. Yes.
Most of you should have heard of as a best-selling book or two.
Well,
right?
So you don't need to read it. You should be able to glance at it and reach a snapshot.
That's right.
Hence the name Gladwell.
Okay, thanks, Jim. The next is from Ricardo. Ricardo is from Brazil. I wonder if he knows the pox story. Anyway, Ricardo asks, hello, TWiV professors. Please read the answers, or at least the winning answer of the Drobo contest, Part 2, Influenza Replication in Bacteria, so all listeners can learn with the winner of the contest. So I'll read today the first one. The winning answer is from Jeff. The question was, we posed, would it be possible to genetically engineer influenza virus so it could replicate in bacteria? Why not? And Jeff writes, first, this depends on the definition of replication, but there are lots of reasons why flu cannot infect and replicate in E. coli. 1, E. coli doesn't have the required receptor proteins to bind flu. 2, E. coli doesn't have a mechanism to internalize flu through its cell wall. 3, E. coli doesn't have a capped mRNAs, which are needed to create flu mRNAs through the cap stealing mechanism. 4, E. coli doesn't have a mechanism to splice flu mRNAs to produce the full complement of flu proteins. This normally takes place in the nucleus of eukaryotic cells. 5, E. coli doesn't have an environment equivalent to the acidic endosome needed to allow escape of the nucleocapsid. 6, E. coli doesn't have ER, Golgi, or enzymes to produce N-linked glycosylated proteins. 7, flu HA and NA proteins would not be directed to the inner membrane of E. coli. Signal sequences wouldn't work. Therefore, virus would not assemble on inner membrane. 8, E. coli doesn't have protease to activate flu HA to make the protein competent to trigger fusion. 9, even if flu mRNAs were made with or without caps, they couldn't be translated in E. coli, no ribosome binding sites. 10, a different codon usage bias may interfere with protein translation. 11, formal methionine would be placed at the N-terminus of flu proteins, may interfere with function. 12, proteins may not fold properly in E. coli, and they may bind to E. coli proteins inappropriately, reducing their effective concentration. For example, basic domains used as nuclear localization signals in mammalian cells may cause problems.
Other than that, it's a go.
Yeah, that's right.
Piece of cake.
However, perhaps we can use a less stringent definition of replication. Perhaps it's possible to try and engineer a system to replicate viral RNAs in E. coli. This would be similar in some aspects to the reverse genetic systems used for flu in mammalian cells. Instead of trying to express all the proteins of flu, we could limit ourselves to the proteins required to replicate a flu genome segment. The flu proteins would need to be under E. coli transcription translation control. For replication, we would need PB1, PB2, PA, and NP proteins. It should also be possible to use plasmids with 2 promoters, opposite orientation, to get one or more of the flu genome strands, opposite of the mRNA, made to act as template for the replicase. Maintaining everything in E. coli is a little tricky. Plasmids with different compatibility groups, plasmid maintenance in E. coli, and antibiotic resistance for selection would be needed. This problem would go away if an in vitro transcription translation, still prokaryotic, were used. One further obstacle to this system is generating the proteins in correct amounts would need a lot more NP than replication enzymes. So that's what we thought was the best one.
Wow.
Yeah, really.
Somebody really wanted a Drobo.
Right. Yes. That was definitely worth a Drobo.
That's fantastic.
I couldn't
when you started to tick them off like that, Vince, I thought I was listening to David Letterman. And the last reason why influenza virus wouldn't infect E. coli is because they hate each other.
Okay, that's enough for email because we're running out of time. Let's do our picks of the week and then close it up. And let's scroll all the way down to the bottom through all this email.
We have a lot of mail.
Rich, what have you got for us today?
Okay, I've been saving this for weeks.
Goodness.
For Dickson.
Uh-oh.
Okay. Some months ago, I watched the documentary film Food, Inc.
Oh, yes.
Seen this?
Oh, I have, sure.
Good. So I include this as a science pick of the week because it's I regard it as technology-based. I think the basic theme here is what has been the impact on basically society in general of advances in technology in the food industry.
Right.
A basic theme of this movie, interestingly, is that in America, the craving for fast food has driven a huge revolution in agro-business. And assorted impacts out from that. The movie had clearly the authors have a bias.
Just a little bit.
Big agro business is bad.
That's right.
But they try and at least superficially, it seems to me, try and present a balanced picture of this. But at several places in the film say that they tried to get this or that big agro business to comment on the on their film, and they refused comment. So you wind up with a more one-sided picture of the thing. It's a very interesting movie and in many ways pretty disturbing.
Yeah.
But I was interested in particular in your comment on this, Dickson.
Yeah. Well, I took my whole class to see it.
Okay.
This was a riveting experience for them because they were not aware of the fact that Monsanto controls the seed industry. I mean, there's only a couple of sources for seeds for commercial production of large crops like soybeans and corn, wheat, rice. That's all been honed down to a very few subspecies of these plants, and highly inbred and homogeneous, so that they're in great danger of extinction because some predator will come along, or some parasite, or some fungus or bacteria or virus will come along and eat them all, because they're all susceptible. It's a clone of seeds, basically. So that was the big take-home lesson that they got from the movie, but also just how prevalent high fructose corn syrup is, and how the agribusiness, and you know what? You can't fault them for it. This is a consumer-driven deal,
right? That and that point comes across.
Yeah,
quite
it does.
strongly
It does, and we demand this food, though. So therefore, okay, fine, we'll just make more for you then. And
right.
All of the consequences of type 2 diabetes and teenage obesity and this sort of thing. So
if we would stop stuffing it in our faces, they would stop making it immediately.
Right.
Here.
And that was the bottom line basically in the movie. They had a nice bullet list of things you can, if you don't like this, here's what you can do.
Exactly.
At the end.
Exactly.
So,
exactly.
At any rate,
thank you, Rich. I deeply appreciate your picking that one.
Well, I'm glad you appreciate it, because I was left. Lots of times, movies like this can be skewed in an unfair fashion.
Yeah, that's right.
And I'm interested to hear you, if I'm hearing you correctly, you would basically endorse this as a reasonable documentary on the issue. Okay,
it is.
So,
it is.
And I'm interested to have that endorsement. Okay, good, great. Well, go watch it.
Did you see this in the theaters, Rich?
No, I got it on Netflix.
Right.
I see. Because I see the DVD is not out yet. It says November 3rd.
Soon.
How did I see it? No, I'm sure I got it on Netflix.
It's already out? Okay. Oh, that's 2008. Okay.
They
haven't updated their website.
Film's been around a while.
We're up on events here.
It's only $9.99.
Tell me, is Hoover still president?
It's also very, very well made.
Putting it in my cart.
The introduction in particular is really good. The way they do the credits and stuff.
No, you're quite right. It's a professional job.
Nice.
Professional job.
See, he was saving that for you for months.
For months.
You mean I haven't been here for months? Come on, Rich.
Yeah,
it's been a while.
It's been a while. No, I agree. It's been a while. I mea culpa. What can I say?
Dickson, are you thinking of a pick? Well,
I am.
Okay.
I am.
Let's get Alan next.
My pick of the week is a website and also an organization called Families Fighting Flu. And I just came across them recently. This group, I think, is the definitive answer to the anti-vaccine campaigners. I wholeheartedly support their mission, and I sincerely hope that I will never qualify to be a member. It's made up of parents whose children all have, almost all of them have two things in common. One is that they were not fully immunized against influenza, and the other is that they're dead.
Right.
Wow.
And it's actually very hard to sit through a presentation by these folks and not be sincerely affected by it. So they, not surprisingly, are big promoters of influenza vaccination.
Sure.
And the striking thing, if you look around on the website and you look at some of the materials that they have, is that most of these are perfectly healthy kids.
Correct.
No particular risk factors, kids running around and happy and healthy, and 2 days later the child is dead.
That's right.
Wow.
One of our faculty members that I'm very friendly with lost a child to flu. So you talk to him about it, and he can give you a real first-hand.
Their argument is not, we make a lot of arguments based on data and logic here, and I think this addresses an entirely separate aspect of the discussion.
That's right.
Yeah, it's good for our TWiV listeners to publicize this because it's got, you have to counter the anti-vax individuals
whose argument is based on emotion.
Yeah.
In the case of the a lot of the anti-vaccine things, there's a false idea that the vaccines are causing. In both cases, they have in common, people are concerned about the health of their children.
That's
Yes.
right.
Okay. But in the case of the, for example, the autism thing, there's no evidence that vaccines cause autism. I'm sympathetic with the fact that these families have really sick kids. That's terrible. But in that case, there's no evidence that the vaccines are causing the problem that they're having. In this case, flu is killing these kids.
Right.
There's no question about that.
Yeah, and they're one or two of the stories. There was one an infant who had received one vaccination against H1N1, but not the second one, the recommended second dose, and died. But the overwhelming majority of them are cases where the kid had not been vaccinated.
Sad.
My view has always been that kids depend on their parents to make the right decisions.
Yes.
They do whatever mom and dad tell them to do.
Right.
Because they
Until
can't
the age of
18 or whatever.
Yeah.
And so you have to make the right decision. It's really unfair if they die because you didn't vaccinate them. So yeah, it's a good one. Dickson, what have you got?
Well, I don't think I've picked this before, so I'll pick it. It's another film called Fuel. Fuel. It's the producer is Josh Tickell, and this is the guy who drove across the country in a minivan that made use of discarded McDonald's cooking oil. Biodiesel is his big deal, and he's a very big fan of vertical farming because, of course, you can grow this stuff, and you can grow it in big, tall buildings if you'd like. So, and it's a well-made film. It's a little bit solipsistic in the sense that he uses his mother and her health problems of growing up in a oil-rich Louisiana as a foil for saying I'm against this because look all the health problems we've caused by it. Of course, he's not wrong. He could have used lots of other examples there too. But
is this also out in theaters?
It just came out in DVD actually, so
right.
I think you can now get it.
I'll get it.
Netflix. You'll like this one too, Rich. It's got a similar kind of bent to it.
Okay, sounds good.
Unfortunately, they don't seem to have kept up with their website domain payments.
Oh dear! You mean it's not there anymore?
Well, there's a message from GoDaddy saying, "Hurry, don't lose your domain," and I don't think that's the trailer for the film.
Oh dear!
So I'm just going to paste in a link to the Wikipedia entry on the film.
Right, or maybe Netflix.
All right, my pick of the week is MIT Open Courseware.
Oh, nice.
It's basically MIT. They post a lot of their course materials in all different fields.
That's true. Lecture
notes, even exams, some audio and some video. It's free, no registration. It's the way all universities should be, really.
Yeah, it's terrific.
But they're not there yet. Now, not everything is up to date. And not everything has video, but it's a great start if you want to see what they're being taught up there.
Wow.
Architecture, engineering, health sciences, humanities, management, science, everything's there. It's really cool.
Fantastic.
And it's all free. Now, Columbia doesn't do this. Neither does Stanford or
We make
Harvard.
money selling our curricula, or at least we think we do.
Now, my lecture notes are free. If you go to iTunes University, you can find my lecture notes there. Very few are available.
Yeah.
As are the ones at MIT. But I think this is a model for all universities.
Do you get any pushback from your Columbia for publishing your lectures?
Not at all. We're all encouraged to publish our lectures at iTunes University or wherever we please. But when you post, the default is to keep it private. And it's not obvious that you have to change it. But I changed it to be public. But most people post and they remain private so no one can see them. So maybe that's really the explanation. I shouldn't be so harsh on Columbia.
Go ahead, be harsh on Columbia.
Can we give our guest a pick too, Vince?
Would you like to make a pick?
Can I take 2 picks?
Yeah, sure.
Actually, to go along the theme of movies, I know we have 2 movies here. There's a movie that just came out, I think, last week called Waiting for Superman. And it's a movie done by the folks who did An Inconvenient Truth. And interestingly, unlike An Inconvenient Truth, which a lot of people basically said had a pretty liberal bent, this actually takes a little more of a turn almost to the right, which is a view of the educational system and a pretty critical view of the unions as being an obstructionist force which is really holding back a lot of certainly, what they say, change for the better in terms of the school systems. And it's hard for me because I'm from a family of educators. My grandmother was a teacher, my mother was a teacher, my sister was a teacher, all in the unions. And unions are, from my perspective, historically have been a force for good. And I think the conflict today with progress in the schools and the unions is something to be deeply examined. So that's the first pick of the week, waiting for Superman. The second pick is around, and I'm actually going through this right now. There's a whole series of articles in Nature this past week, which is "Can Science Feed the World?" And I'm in the process of reading this, but my skeptical view is just wanting to make sure there's a balance between sort of what is big agribusiness and what is sort of the forces arrayed against big agribusiness, be it universities or be it folks involved in sort of the almost the microfinance side of things. But bottom line is this is a big issue and it'll become more significant, obviously, not just as the world's population grows, but as people consume more, as each person consumes more. And so anyhow, it's about a 20-page section within Nature this past week.
Yeah, I'm looking for it here on the front page, but I'm not seeing
I just popped a link into
it's the July 29th
good. Here
issue.
we go.
Excellent.
Yeah.
Can Science Feed the World with Dickson Despommier.
Depends on whose science, I guess, in this sense.
Well, Dickson, you would say vertical farming is science, right?
I'm saying that science has the answers to the questions. What was your question? Can I make a lot of money selling corn and soybeans? Of course you can.
You can get carried away with these picks, by the way. I can also say Coogan's, which is for lunch.
It's a local restaurant that's got international reputation.
Yes.
We have pretty broad tolerance for picks, right?
Yeah, we do.
All right, that's great. Thanks, everybody. And, of course, as always, there are many ways you can listen to TWiV. You can go to the iTunes store. It's free there. You can go to the Zune Marketplace. And if you do, why don't you subscribe so you get episodes automatically? And if you're new to TWiV, leave a comment on iTunes. It helps us to stay on the front page of the Medicine Podcast directory. You can also listen to TWiV with Stitcher Radio, which is a free app for your smartphone, for your iPhone, for your iPod Touch, iPad, BlackBerry, Palm, or any Google Android-driven device. So just search for Stitcher Radio or go to bit.ly/StitchTWiV. Of course, you can always go to twiv.tv and play or download the episodes there. We have the complete archive as well as show notes. TWiV is part of microbeworld.org, sciencepodcasters.org, and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. And don't forget to buy your TWiV merchandise and be a supporter at cafepress.com/TWiV. Send us your questions and comments to twiv@twiv.tv. Dickson Despommier, good to see you again.
Same here, Vince. Good to see you.
Hope to see you soon again.
Good to hear from the guys.
You bet. Alan is at alandove.com. And the latest is, this is a great post, which I'm waiting to load. Come on, Internet.
Oh, my site is slow today.
Too many visitors, no doubt.
Yeah.
Crash the site 3 times.
Look at this. It's not loading. Here we go. Why does it hurt when I pee?
Right.
Go there and read it. I won't tell you about
That's
it.
Alan's post of the day. I like that.
Yeah,
Yes.
just another
I
Alan
like
Dove. All right, thanks, Alan. And Rich Condit is at UF Gainesville, home of the Fighting Gators.
Hey.
Indeed.
Good to have you again.
Always a good time. I think that's Alan's line, but I like it.
It's a good line. And our guest, Michael Zygmunt from the New York Academy of Sciences. Hope you enjoyed it.
Thanks, and I did, and I was glad to be in both an in-studio guest and an in-guest studio, so I appreciate it.
Excellent.
Hey, wait a minute, Alan. P is P-E-E, not P-E-A.
Read the
Oh, no,
post.
wait a minute.
You got to read it, man.
Come on.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.